Here's what everyone thought they knew by 2017. A team from the University of Tübingen and the Max Planck Institute in Jena had successfully recovered mitochondrial genomes from 90 mummified individuals at a site called Abu Sir el-Malek in Middle Egypt. They also extracted partial nuclear data from 3 of those individuals. Headlines declared that scientists had finally cracked the code. "We know who the ancient Egyptians were," the article said. "The genome spoke. The answer was in." If you've ever actually wondered where you come from, not the family story, the real biological answer, subscribe. Because what this field has found in the last 12 months goes deeper than most people are willing to sit with. But here's what almost nobody checked. Those 90 individuals came from one site, in Middle Egypt. The youngest of them was born when the Great Pyramid at Giza was already 1,200 years old. The people who built that pyramid, the architects of the first dynasty, the engineers of the earliest hieroglyphic systems, the founders of the civilization that every subsequent dynasty inherited, were genetically invisible. Every headline about ancient Egyptian DNA was built on a dataset that couldn't touch the civilization it claimed to describe. Then, in 1902, a British excavation team at a limestone hillside 265 kilometers south of Cairo pried open a ceramic pot and found a skeleton inside. They cataloged it, shipped it to Liverpool, shelved it in a museum. For 123 years, nobody ran its DNA. Nobody thought to. What they had, without knowing it, was the one burial type that had accidentally defeated Egypt's climate. No mummification chemicals, no humidity cycling, just sealed clay, carved stone, and perfect preservation. The missing variable had been sitting in a drawer in England since before the Wright brothers flew. World Museum, Liverpool, 2022. A researcher named Adeline Morez-Jacobs, a visiting fellow at Liverpool John Moores University, is standing in front of a storage box. Inside, a partial skeleton donated to a British excavation committee by the Egyptian Antiquities Service in 1902, under British administration of Egypt. The man inside the box has been dead for approximately 4,700 years. He was never mummified. And that, as it turned out, was the accident that saved him. Morez-Jacobs, working alongside bioarchaeologist Joel Irish and ancient genomics lab head Pontus Skoglund at the Francis Crick Institute in London, extracted DNA from the root of one of the man's teeth. Using shotgun sequencing, a method that reads all the DNA present and filters for human material, they achieved 2 times coverage of his whole genome. The highest resolution ever reached for any Egyptian individual. The first complete nuclear genome from ancient Egypt. Radiocarbon dating placed his death between 2855 and 2570 BCE. The early dynastic to Old Kingdom transition. Within a century of the construction of the Great Pyramid, he was the civilization, not a record of it. The thing itself. 40 years after Pabo walked away empty, the genome that broke Egypt open didn't come from a mummy. It came from a man who died before mummification was standard practice, folded into a ceramic pot inside a limestone hillside, and the stable, cool microclimate of the rock did what 4 decades of laboratory technology could not. The isotope analysis of his teeth confirmed he was born and raised in the Nile Valley. His ancestry, though, had not been local for generations. Statistical modeling of his genome required 2 source populations to explain what was there. Roughly 80% aligned with Neolithic North African populations from ancient Morocco, people who lived as far back as 4,780 BCE. The remaining 20% traced to the Eastern Fertile Crescent. Neolithic Mesopotamia, modern-day Iraq. His bones told a second story. His seat bones were expanded from sustained seated labor. His right arm showed evidence of repetitive lateral motion. His right foot alone had significant arthritis. He died somewhere between 44 and 64 years old, and he was buried in a high-status rock-cut tomb. The man who lived at the dawn of pharaonic Egypt bore in his skeleton the marks of a working life, and was interred as someone important. That contradiction has not been resolved. But here's what stopped me when I first read this. The 80% North African ancestry in his genome doesn't trace to Egypt itself. It traces to ancient Morocco. to people who lived 800 years before he was born. The ancestry of the man who lived alongside the first pyramids had been forming on the other side of the Sahara for centuries before he existed. The Sahara wasn't a barrier, it was a corridor, and by his lifetime, it was closing. Before July 2025, the standard explanation for why early Egyptian and Mesopotamian artifacts look so similar, the cylinder seals, the architectural niching, the specific pottery forms appearing in both regions around 3200 BCE, was diffusion. Ideas traveled, trade routes carried objects, the people themselves largely stayed put. This wasn't a fringe interpretation, it was the archaeological consensus, held for decades, published in textbooks, taught as settled history. Joel Irish, the bioarchaeologist at Liverpool John Moores University, who co-authored the 2025 Nature paper, put it plainly when the results came in. The Nuweirat genome provided the first genetic confirmation that the exchange between Egypt and West Asia wasn't just cultural. People moved. Not just pottery. What that means precisely. The 20% Mesopotamian component in that genome doesn't represent a man who traveled from Iraq. The isotopes in his teeth show he was born in the Nile Valley. His Mesopotamian ancestry had arrived at least one generation before him. Someone made the crossing before he existed. We don't know when. We don't know how many. We don't know what they called themselves. What we know is that by the time writing existed to record them, they were already Egyptian. The arrival had been absorbed before history could name it. Everything I just told you? Hold on to it, because it's about to mean something completely different. Every museum exhibit showing Egyptian and Mesopotamian objects side by side. Every textbook separating these civilizations as distinct entities that influenced each other. Technically correct, and functionally incomplete. The exchange left DNA. The DNA was in a pot in Liverpool. But here's where the science demands honesty. Barry Kemp, the Egyptologist at Cambridge who has spent decades studying ancient Egyptian population history, has pointed out that DNA studies can only yield firm conclusions if samples represent a broad geographic and chronological range. The Nuweirat genome is one person. He may represent an entire migration wave. He may be an exception. The science is specific about exactly one thing: one genome, one man, one pot. What he tells us about the millions who built and populated ancient Egypt remains, right now, an open question. And that creates a specific problem. Two peer-reviewed studies, both published in major journals, both methodologically sound, producing different pictures of ancient Egyptian ancestry. The 2017 Abusir el-Malek study pointing toward Near Eastern and European affinities. The 2025 Nuweirat genome pointing toward a majority North African Neolithic foundation. Both real findings, both limited by sample. And the 1400 years between them? The genetic gap separating who we thought the ancient Egyptians were from who the evidence now suggests they might have been, still largely unread. 1400 years is a long time to be certain. Abusir el-Malik, Middle Egypt. The 2017 study sampled every mummy they could access at one archaeological site. One site, in Middle Egypt. Upper Egypt, the region geographically closest to sub-Saharan African populations, was not represented at all. The Old Kingdom was not represented. And the study's lead researchers, Verena Schünemann at the University of Tübingen and Wolfgang Hack at the Max Planck Institute in Jena, acknowledged that limitation explicitly inside the paper. The field cited it anyway, as the definitive statement on ancient Egyptian ancestry. And yet the data said something the headlines refused to. By 2022, archaeologist Danielle Candelora had put the vulnerabilities on formal record. New and untested sampling methods, a small sample from a single site, and problematic comparative population data. By 2023, historian Christopher Ehret at UCLA called the interpretation biased, arguing specifically that the study had dismissed a sub-Saharan component in ancient Egyptians using a geographic criterion that doesn't hold when you look at the actual Horn of Africa ancestry roots. In the same year, historians William Stiebling and Susan Helft noted that the study couldn't represent Egyptians from Upper Egypt at all. people geographically closer to sub-Saharan populations who may have left no readable mummies because the heat of the Egyptian south destroys DNA in ways that the cooler north does not. Their absence from the dataset wasn't random, it was structural. Now, the 2017 study did not misrepresent what it found. The researchers found exactly what they found. What happened next? The citation cascade, the popular science articles, the summaries declaring that ancient Egyptians were from the Near East was not what the study concluded. The study said, these people, at this site, in these centuries, showed these genetic affinities. Everything beyond that was extrapolation built by the secondary literature, not by the science itself. Actually, hold on. Before we go further, I want to ask you something real. Drop it in the comments. What do you think is more dangerous? A study that gets the data wrong, or a field that gets the data right but frames it beyond what the data actually permits? I mean it, I read these. Because your answer might be more connected to what I'm about to say than you'd think. Stay with me, because this next part is the reason this video exists. The most cited genetic analysis of ancient Egyptians is still the 2017 study. It has not been retracted, it has not been superseded, and yet everything it could access, geographically, temporally, methodologically, placed it in a context that the 2025 Old Kingdom genome now reframes entirely. Whether ancient Egyptians were closer to Near Easterners than modern Egyptians is true for the populations that built the civilization. The people who lived before 1388 BCE remains, as of today, a question answered by exactly one complete genome published less than a year ago. One genome, one family, one result that nobody fully explained. Cairo, 2010. The Egyptian National Research Center and Cairo University's Faculty of Medicine have built two of the most controlled ancient DNA laboratories in the world, set up specifically to analyze the royal mummies of the 18th dynasty. Dr. Yahia Gad, leading the research for the National Research Center, enforces a contamination protocol rigorous enough that every female mummy is tested for Y chromosome DNA to catch any trace of male lab worker contamination. Every female mummy comes back negative. The science is airtight. In 2020, Gad and co-author Zahi Hawass published the results in Human Molecular Genetics. The Y chromosome haplogroup of Tutankhamun's family, Amenhotep III, Akhenaten, and Tutankhamun himself, is R1b. The maternal haplogroup, traced from Tutankhamun's great-grandmother Thuya through his still unidentified mother, is K, both confirmed under controlled conditions, both officially unresolved at the level of subclade. The specific downstream branch of R1b was not published with the full resolution needed to place it precisely on a haplogroup tree. What they found next changed how this story would be told for years. Here's what the study did not explain, because it couldn't. Haplogroup R1b appears in under 1% of modern Egyptians. It is the dominant haplogroup of Western Europe, carried by more than 60% of French men, more than 50% of Spanish men, more than 80% of Irish men. The researchers stated explicitly that these findings said nothing about ethnic origin. A haplogroup is one paternal line in a genome of thousands of lines. It is not an identity document. But the subclade determination, the data that would tell you which branch of R1b, has not appeared in any peer-reviewed paper at sufficient resolution to be definitive. This is not suppression. This is the reality of working with degraded ancient material. Getting subclade precision from ancient royal mummy DNA requires coverage levels that material this old often can't provide. The haplogroup assignment is robust. The subclade is genuinely unknown. The problem came from outside the lab. A Swiss genealogy company issued a press release based on a blurry screen grab from a television documentary, screen grab resolution, not sequencing data, and claimed it proved Tutankhamun's haplogroup subclade. The claim spread globally. It was used as racial evidence by extremist communities online. Egyptian scientists refuted it. The refutation didn't travel nearly as far as the original claim. The specific downstream branch of R1b, carried by the family that built Karnak, that produced Akhenaten's experiment in monotheistic state religion, that commissioned the Amarna letters, it is officially inconclusive. Not suppressed, inconclusive. And what fills an inconclusive finding is almost always ideology, not evidence. R1b in under 1% of modern Egyptians. And yet the Egypt Genome Project, which sequenced 1,024 living Egyptians last month, identified a distinct 18.5% genetic component found in no other population's database anywhere in the world. If R1b in the royal family represents an ancient lineage now nearly absent from the living population, and that living population carries 17 million genetic variants invisible to global science, the question isn't what the ancient royals carried. The question is, what else is in that genome that the world's scientific infrastructure has never properly looked at? Cairo, April 6, 2026. Dr. Khaled Amir of the Egyptian Center for Research and Regenerative Medicine and Dr. Ahmed Mustafa of the American University in Cairo announced the publication of the Egypt Genome Project, the first large-scale whole genome sequencing study of modern Egyptians. 1,024 individuals, 21 of Egypt's 27 governorates, Upper Egypt, Lower Egypt, the Delta, the Suez Canal region. The first time the genetic diversity of a living population of 100 million people has been systematically put on record. The study identified 51.3 million genetic variants across the participants. Of those, 17.1 million were absent from dbSNP, the global variant database maintained by the National Center for Biotechnology Information in the United States. A third of the documented Egyptian genome's variation simply does not exist in the reference databases that doctors worldwide use to interpret genetic risk, predict disease, or calibrate pharmaceutical response. Modern Egyptians have been carrying a third of their genetic diversity in a biological silence that global science has never registered. This isn't just ancient history, this is in your DNA right now. If your ancestry intersects with North Africa, the Levant, or any part of the migration corridors that run through Egypt, some of what your genome carries may be variant data that no database in the world has a reference for. Not rare variants, not damaged data, simply undocumented. The world's medical infrastructure was never built to look for you. The study also confirms something that closes a loop opened 4,800 years ago. A distinct 18.5% genetic component in modern Egyptians, likely the continuity signal connecting the Old Kingdom man from Nuweirat, folded in his ceramic pot, to the 100 million people living above those tombs today. The genetic bridge across nearly 5 millennia exists. It is specific. It has been documented. The ancient and the living are connected, not as metaphor, but in base pairs. Allele frequency comparisons across 6.5 million shared variants showed the strongest genetic concordance between Egyptians and Middle Eastern populations, a correlation so high it confirmed that the identity built in the Nile Valley over millennia has been continuous enough to survive every conquest, every dynasty, every empire that crossed the Sinai. The answer had arrived. And then, the orange banner at the top of the document, the bioRxiv preprint stripe. The EGP1K paper, as of the date of this episode, has not yet completed full peer review. The 17 million novel variants have no comparative reference population anywhere. They cannot be validated against a second Egyptian Genome Project of equivalent scale, because no second project of that scale exists. The answer arrived. And in arriving, it revealed that the infrastructure needed to use the answer does not yet exist. The gap the science just closed opened 3 new ones. And what lives inside those gaps, undocumented, uncharacterized, present in every Egyptian alive today, is the question the field is now racing to answer. No category. And no category means no treatment. A physician in Cairo opens a genetic risk report for a patient. 34 years old, asymptomatic. No family history of cardiovascular disease. The report, generated by a polygenic risk score algorithm built from European genome data, classifies the patient as high risk for stroke. The algorithm isn't broken. It calculated exactly what it was designed to calculate. The problem is that it has no reference for what a typical Egyptian genome looks like. It is comparing this patient to the wrong population. And as the EGP1K study revealed in April 2026, it is doing this to almost everyone it tests. The researchers applied European-derived polygenic risk models to all 1,024 participants. The results? 83.3% of Egyptians classified as high risk for stroke, 76.4% for chronic kidney disease, 72.9% for gout, These models are built to flag the top 10% of any population. When 83% of a population is flagged, the model has not found a health crisis. The model has proven that it does not know what it is measuring. The genome databases built over 40 years to decode human disease are so overwhelmingly European that when their risk algorithms were applied to 1,024 living Egyptians in April 2026, they flagged 83% as being at imminent risk of stroke. A number so impossible it reveals not a health emergency, but a scientific system that built the map of all human genetics without ever properly sequencing one of civilization's oldest continuous populations. There are three things this finding tells us. The third one is the one that changes everything. The first, this is not a problem specific to Egypt. The same European-derived reference data that mislabeled 83% of Egyptians is the same infrastructure used by genetic testing companies across the United States, the UK, and throughout the Middle East for populations whose ancestry intersects, in whole or in part, with the migration corridors running through the Nile Valley. The second, those 17 million undocumented variants belong not just to Egyptians, but to everyone whose biological history crosses that ground. And the third, Egypt's Ministry of Health has already confirmed it will develop what they called 100% Egyptian personalized genetic tests to replace the European-derived models currently in clinical use. The error is being corrected right now, which tells you everything about how long it was running uncorrected. The study also confirmed that 9.1% of the Egyptian population carries the gene for familial Mediterranean fever, generating approximately 6,600 affected births every year. Many of those births happen before the diagnosis is made. The European risk models didn't predict it. They weren't built to look for it. The same institutional blind spot that left ancient Egyptian DNA unstudied for 40 years is now operating in diagnostic software, in pharmaceutical trials, in cancer screening, in the bodies of living people. The Nuweirat man's 4,800-year-old genome is not a curiosity. It is evidence that an entire branch of human diversity has been running outside the map for the entire era of modern genetics. And the Nile Valley, the corridor through which humanity first left Africa, is still waiting to be fully read. What was read before it could be read? The nameless people. Somewhere in the Eastern Fertile Crescent between 3,500 and 3000 BCE, a community of people, their names unrecorded, their language lost before anyone wrote it down, their faces unimaginable, begin moving west. Not as conquerors, not as a documented trading party. The isotopes in the Nuweirat man's teeth show he was born in the Nile Valley. His Mesopotamian ancestry had arrived at least one generation before him. Someone made the crossing before he existed. We don't know their name. We don't know what they called themselves. We don't know whether they thought of themselves as leaving somewhere or arriving somewhere or simply moving the way people move when conditions make staying harder than going. The Nuweirat study places the likely arrival window at the pre-dynastic to early dynastic transition, roughly 3,200 to 2,900 BCE, a period archaeologists have long noted for a sudden acceleration of complexity in Egypt. The pottery wheel, cylinder seals, new architectural techniques. The genetic data now confirms what the artifacts only implied. Some of the people who carried these technologies didn't send them ahead. Morez Jacobs and her co-authors are careful. The word circumstantial appears deliberately in the paper. The bones suggest a craft. The genome confirms a crossing. Together they tell a story neither could tell alone. The reframe. These people didn't arrive as immigrants in any sense history could record. They arrived early enough that by the time writing existed to name them, their grandchildren were already Egyptian. The arrival was absorbed before the tools to document it existed. Every account that treats ancient Egypt as a purely indigenous African civilization is missing the 20%. Every account that treats the Mesopotamian connection as foreign influence misses the 80%. Egypt was not a product of either world. It was the specific place where these two worlds became one person, folded into a ceramic pot, inside a limestone hillside, holding a genome that took 4,800 years to read. And the science demands this honesty. The 20% Mesopotamian component exists in exactly one Egyptian genome. The researchers who published it are explicit. This individual may not represent the broader Old Kingdom population. The sample size is 1. The finding is verified. Its population-level meaning is genuinely unknown. The unnamed people who crossed from the Fertile Crescent to the Nile before the pyramids existed may have been many. They may have been few. They left no monument that carries their name, no inscription, no text. Only a fraction of a genome in one man's tooth, in a pot, in a museum in Liverpool. They never had a name, but they had children. Their children are the answer to a question that wasn't asked until 40 years ago. A study cited in the Egypt Genome Project identified modern Egyptians as the African population whose genomes and haplotype frequencies most closely resemble those of non-African populations globally. Not closest to Europeans specifically, closest to all non-African populations as a group. Think about what that means. The people living above those limestone tombs, above the ceramic pot, above the unnamed Mesopotamian migrants, above every dynasty that built and fell and was rebuilt, carry in their bodies the clearest surviving genetic signal of the moment humanity first crossed from Africa into the rest of the world. The out-of-Africa migration route through the Sinai Peninsula is not an archaeological theory inferred from stone tools and trade goods. It is written in the genome of every living Egyptian. If you are not of African ancestry, your non-African genetic heritage crossed through this corridor, not near it, through the specific ground where these people have lived continuously for 10,000 years. The people who live above those tombs are not simply the inheritors of an ancient civilization. They are the living record of the moment your own ancestors left the continent where the human story began. And here is what that means right now in 2026. The Egypt Genome Project identified 17.1 million genetic variants in living Egyptians that exist in no global database. No doctor working from standard reference data, no genetic testing company operating from established population references, no pharmaceutical researcher using current databases can fully interpret what those variants do. They may affect disease risk. They may change how a drug metabolizes in a body. They may carry adaptive signatures from the Green Sahara period, from the Mesopotamian integration, from any of the dozen migrations the Nile Valley has absorbed and transformed across 10,000 years. If your ancestry intersects with the populations that moved through Egypt, and for most of humanity the out-of-Africa route makes that intersection possible, some of what your genome carries may be running entirely outside the map that medicine built to read it. The next episode in this series goes deeper into a connected corridor, findings more recent and more unsettling than anything in this one. Subscribe and it comes directly to you. The Nuweirat man was buried in a ceramic pot in a limestone hillside 4,800 years ago. He was excavated in 1902. His genome was read in 2025. The databases needed to fully interpret what his genome says about the 100 million people living above his tomb do not yet exist. We know he was here. We know what he carried. We know that 20% of him came from the Fertile Crescent before history could record the crossing. We know that 80% came from a Saharan North Africa that no longer looks anything like it did when his ancestors lived there. We know that something of both those worlds survived, absorbed, transformed, continuous, into the living people who gave blood samples in clinics across 21 governorates and waited for results that the world's databases weren't yet built to return. That work is happening right now, in Cairo, in London, in Leipzig. And when the answer arrives, it will change what every human being understands about the ground beneath their feet.